Imagine this, a world where farmers can grow crops that resist deadly pests without spraying a single drop of pesticide. A world where rice can survive prolonged floods and where cows naturally produce more nutritious milk. Sounds like science fiction, right? Well, it's not fiction, it's science. And the heart of all of it is a molecule you have heard of DNA. So welcome everyone, my name is Kant Stavan, I'm Sulihon. We, Group 5, are talking with you on a journey through nucleic acids, what they are, how they work, and how they rewriting the future agriculture. So let's get into it. So before we talk about crops and biotechnology, we need to understand the molecule behind it all, deoxyribonucleic acid or DNA. So DNA belongs to a family biological molecules called nucleic acids. So these are informational macromolecules. They store and transmit the genetic blueprint of living organism.
So what exactly makes up DNA? DNA is a polymer, meaning it's built from repeating units called nucleotides. Each nucleotide has 3 parts. 1. A 5-carbon deoxyribose sugar. 2. A phosphate group. And 3. The most important part, a nitrogenous base. These bases comes from 4 types: adenine, thymine, guanine, and cytosine, as simply as A, T, G, and C. Now, here's where it gets elegant. DNA is a double helix, 2 strands twisted together like spiral staircase, and those strands are held together by specific base pairing. According to Chargaff's rule, adenine always pair with thymine, and guanine always pair with cytosine. Gene expression happen in 2 steps. First, transcription. Inside the nucleus, an enzyme called RNA polymerase reads that messenger RNA or mRNA. This mRNA then travels out of the nucleus. Second, the translation. In the cytoplasm, the ribosome reads the mRNA into groups of 3 bases called codons. Each codon corresponds to a specific amino acids. These amino acids chain together to form a protein. And the protein, they do everything. They build tissues, catalyze reaction, and determine traits. This entire process from DNA to RNA to protein determines whether the plant grows tall, resists disease, or produce more grain.
First is the CRISPR-Cas9. CRISPR stands for clustering regularly, interface, short palindromic repeats. It's a gene editing tools that uses a guide RNA to locate a specific DNA sequence, like a GPS for the genome, and the protein called Cas9 to cut it precisely in agriculture. CRISPR has been used to develop with resistant to powdery mildew, tomatoes with longer shelf life, and drought tolerance crops. A landmark 2018 study by C. et al. In the journey, Nature Plants demonstrate how CRISPR editing of yields relating gene of maize increase grain size and production. This study is cited in our video description. Now, here's where everything we just learned gets incredibly exciting, because scientists have learned how to engineer and harness nucleic acid to transport agriculture. Let's look at 3 major applications.
Next, genetically modified organisms, or GMOs. Unlike CRISPR, which edits existing genes, GMOs involves inserting the gene from one organism into another. The classic example, BT crops plants engineered with a gene from the bacterium Bacillus, producing genesis that produce a natural insecticidal protein. The result, reduced pesticide use, lower cost for farmers, and higher yields. According to the International Service of Acquisition of Agri-Biotech Application, or ECAA, over 190 million hectares of GMO crops are planted globally each year. Number 3. RNA interference or RNAi. This is a biological mechanism where small RNA molecules silence specific genes. Scientists are now digesting dsRNA or double-stranded RNA, sprays that can target and express genes in specific pests without harming the other organism. For example, researchers have developed RNAi sprays targeting the Colorado potato beetle, one of the most destructive potato pests in the world, silencing its vital genes and killing it without chemical pesticides. The precision of RNA is what makes the approach of revolutionary.
Before we wrap up, we want to be honest with you. These technologies are powerful, but they are not without controversy. So, as a future agricultural scientist, we believe it's our responsibility to think critically, not just technically. So, let's talk about benefits, risks, and ethics. On the benefit side, nucleic acid-based technology can dramatically increase food security, reduce chemical inputs, improve nutritional quality, and helps crops to adapt climate change. For a country like the Philippines, where rice is life, food-tolerant and pest-resistant varieties can protect millions of farming families.
But the risks are real too. Gene flow where modified genes spread to wild plant relatives could disrupt ecosystem. There are concerns about monopolization of seeds by biotech corporation, limiting access for smallholder farmers, and long-term effect of some GMOs on human health are still being studied. These are not reason to stop science, but they are reason to do science carefully, transparently, and with a community input. Science without ethics is just technology, but science with ethics, that progress.